Hello, welcome back to part 2 of the nervous system. Last time we spent our time discussing general concepts of the nervous system, how it's all integrated together, and then we spent a good deal of time talking about the structure and function of the neuron. Now in part 2, we're going to be talking mostly about glial cells, or the support cells to the neurons. So these are also sometimes referred to as neuroglia. You will find glial cells in both the central nervous system and the peripheral nervous system. They're much smaller than neurons. They don't stain nearly so darkly in histological samples. And these cells are capable of mitosis. In fact, as I mentioned earlier, most types of brain tumors or brain cancers are derived from glial cells, but not all of them. Their job is to protect and help nourish the neurons, help them in their function. There are many more glial cells than are neurons, right? So there's a much greater number of them composed to neurons. So this picture is showing us the various different types of glial cells in the central nervous system, and we're going to find that there's some different support cells between the central and peripheral nervous system. So this is a cross-section of the brain, or a coronal slice to the brain. We look here, we can see our neurons, right? Our big multipolar neurons. The surrounding cells, these green ones, are called astrocytes, and astrocytes are sort of star-shaped, and which is the term astrocyte, or astro comes from. These are multi-branch cells that are going to help connect neurons to blood vessels. So astrocytes will provide actual physical structural support for the neuron, and they're going to serve as an intermediary between the vasculature and the cells. This is a bit of a unique arrangement. In most other parts of the body, you don't have an intermediary between some random cell and the blood vessel. We'll talk more about this in a minute, but really astrocyte is one of the primary components of what's called the blood-brain barrier. We'll talk about in just a moment. Now, this other cell over here looks, I don't know, like a little spider-like. This is called a microglial cell. This is actually a cell of your immune system, and it is there to help keep this tissue safe. Now, for reasons that we're going to get into in a second, most of the rest of your immune system cannot really gain access to the brain. And so it's the microglia's job to help keep any unwanted pathogens, viruses, bacteria, protozoa, whatever, out of this area. So these guys are indeed quite important. Now, this cell here is called an oligodendrocyte. There's that dendro again. So dendro, meaning branch, cyte means cell. Oligo, when you see oligo means many. So an oligodendrocyte is literally a cell with many branches. It's different than neurons, but it still has lots of branching appendages. In this case, an oligodendrocyte is a cell, it is a glial cell in the central nervous system, which is responsible for myelinating the axons of neurons. So oligodendrocytes are only found in the central nervous system, and their job is to myelinate multiple sections on multiple different axons. This is different than the cell responsible for myelination in the peripheral nervous system. It's a little bit different. We'll get to that in a second. But here, an oligodendrocyte has many branches. It can myelinate multiple sections on the same neuron or even on different neurons. Last but not least over here, we have ependymal cells or ependymal cells. These cells are what's going to create the cerebrospinal fluid, the CSF, that is inside our brain, or actually our brain floats in cerebrospinal fluid. So this is just the 4 types: astrocytes, ependymal cells, microglia, and oligodendrocytes. Support the neurons, provide structure for the tissue, and help form the blood-brain barrier. Ependymal cells will help create CSF, cerebrospinal fluid. Microglia will remove any cellular debris or help keep pathogens away. And oligodendrocytes are cells from myelination in the central nervous system. So let's talk about astrocytes first. They're by far the most abundant type of glial cell in the central nervous system, and they help form this blood-brain barrier. So just so we can better understand what this is, let me talk a little bit about the blood-brain barrier. Normally in your body, capillaries leak. They leak fluid. They don't leak cells, but they leak fluid. And this is the normal thing because the fluid inside our blood, the plasma, when it leaks out of the capillary, it becomes interstitial fluid, and that helps carry dissolved oxygen and nutrients and carry away waste. So we want this to happen in most of the rest of the body. But that leakiness means that you have less control over what leaves the blood vessels and enters the tissue, right? That is a danger in the central nervous system because remember, neurons are non-mitotic, right? They're not going to be replaced. So if a neuron dies, it's gone. And also remember, neurons are very delicate. So they're very valuable, very delicate, can't be replaced. So there's this blood-brain barrier, which prevents material from leaking uncontrolled out of the blood vessels, at least compared to the rest of the body. So I'm going to get a little bit ahead of ourselves. This will make more sense once we get into the vasculature, but just so you understand exactly what this is, blood vessels, the capillaries in our central nervous system are special. They do not leak, right? So they're special blood vessels which don't leak. However, our tissues, our neurons still need nutrients. They still need oxygen. They have to get rid of carbon dioxide. They have to get rid of waste. So now we have a middleman, the astrocyte. Say you want to get nutrients out of the blood supply. Normally, in other parts of the body, it would simply just leak out the tube and then bathe the cells of the tissue and everything's going to be just fine. But we don't want that happening here because we want to keep toxins or unwanted materials or pathogens away from this very critical area. So the way that the astrocytes perform this job is anything that is inside the blood you want to deliver to the neuron, it's going to have to be exported out an endothelial cell, right? The simple squamous epithelium you see right here. It will then have to pass into the plasma membrane of the glial cell, go from one end of the astrocyte to the other end of the astrocyte, be transported across the plasma membrane of the astrocyte, and then transported across the plasma membrane of the neuron. Right. So you have lots of membranes and lots of structures to pass. That gives you a lot of opportunities to control what's going to pass through those semi-permeable membranes. So the purpose of this is to act as a gatekeeper to ensure that only the materials that you're absolutely sure of are going to get from the circulatory system to the neuron. Right. And that's what the purpose of the blood-brain barrier is. So it's really kind of a double-edged sword. It's a good thing and it's a bad thing. If you go and eat a bad oyster, right, you might absorb that and maybe not all the toxins get removed by your liver. And if that hits your circulatory system, right, what cells are by far and away the most fragile, right? If a tiny amount of toxin gets in there and kills half your brain cells, right, you're done. Right. So in order to prevent that, you have the blood-brain barrier to keep even those low levels of toxins, which would be a big problem for the nerves, but are not such a big problem in the rest of your body. Right. The rest of your body can deal with that, but the neurons can't. So they have that extra level of protection. Now, downsides. The way that your immune system tends to get to where it needs to be, it will use blood vessels as a transportation, it will use them as a highway, as a transportation mechanism, right? Because these blood vessels do not leak, it's really hard for our immune cells to pass through these blood vessels. So it's really hard for your immune system to get to your brain and protect that tissue if an infection does develop. The only thing you got are these microglia to help keep you safe. Most of the rest of the immune system cannot get into your brain because it's what's called privileged. Basically, the blood-brain barrier will prevent most of the rest of your immune system from entering and protecting your brain. This is just one of the reasons why something like encephalitis or meningitis, right, inflammation of the brain caused by viruses or bacteria, right, these diseases can develop very, very rapidly, right, because if something gains access to your brain, and it's not supposed to, right? You can go from, man, I don't feel good, I have a headache, to dead in just a few hours, because you have very high value cells, very easily damaged. You can't get the immune system there. And if you get a raging infection, it can do a tremendous amount of damage in a very short period of time. So possible downsides, it's a little bit harder for the rest of your immune system to come to the defense of this tissue. And also, certain drugs are not going to pass the blood-brain barrier because it's not going to make it past all those selectively permeable membranes, right? So a lot of medicine you take might be really great to help fight off an infection in your brain, but it's not going to do any good if you can't get past that blood-brain barrier. So it's kind of an important physiological concept. I know this is not a physio class, so I'm not going to like rigorously test you on the blood-brain barrier, but I felt I kind of need to explain a little bit about it, so you will give you some relevance to know that astrocytes make up part of this blood-brain barrier, right? The job of the astrocyte is to actively take materials from the circulatory system and then hand it off to the neuron. Because normally in the rest of the body, stuff just leaks out of the blood vessels and bathes all the surrounding cells. It's very easy, but it's not particularly controlled, right? The job of the astrocyte, structurally support the neuron and support it with nutrients by acting as a middleman between the blood vessels, the capillaries, and the neurons themselves. Okay. Astrocytes are also important for sort of regulating the fluid composition in these tissues, right? Acting as that middleman. Helps regulate synaptic transmission. It just has to do with keeping the cell healthy, and you can learn more about that in physio. Structural framework that I talked about. Astrocytes can also help repair damaged neurons, and they actually play a role in neuronal development, but that's really outside the context of this class. So here's just a blow-up image of our neuron. It's being myelinated by oligodendrocytes. Here's our astrocyte serving as a middleman between a capillary and the CNS and the neuron. And these little perivascular feet are going to connect blood vessel to neuron. Now, ependymal cells, these are actually a ciliated cuboidal epithelium that lines the ventricles of the brain. Now, the ventricle of the brain, you actually have holes in your brain, empty spaces, and that's good, and they're called ventricles. And they're filled with fluid called cerebrospinal fluid. In fact, inside the ventricles, you have structures called choroid plexuses, and inside a choroid plexus, you will find ependymal cells. So ependymal cells take blood and turn it into cerebrospinal fluid. You find ependymal cells inside the choroid plexus. Choroid plexuses are found inside the ventricles, which are spaces or cavities in the brain. So if we go back to this image here, you see this area in blue? This is an open space. This is a cavity inside the brain. And lining that structure here, you see ependymal cells creating cerebrospinal fluid from blood. Now, microglia, small cells that are mobile, right? These are part of your immune system. They can crawl around, look for cellular debris, look for damaged cells, look for any unwanted material, pathogens, right? And just gobbles them up and destroys it and just keeps the tissue clean and healthy. Right, these little spider-looking guys. And then finally, we have the oligodendrocytes. These are only found in the central nervous system, and their job is to wrap around one individual portion of an axon, and then they can wrap around a second, or perhaps even attach to multiple axons of different neurons and provide myelination. So what myelination is, think of it, at least at this moment, as insulation around the wire of the axon. What it's really going to do is it's going to help the action potentials travel faster and more efficiently. So just think of it as insulation around a wire. It helps conduct the current better. Actually, not exactly true, but it will help increase the efficiency, and it will increase the speed of an action potential of a neuron. Right. Remember, oligodendrocytes means many branched cell. It will attach to multiple axons and myelinate multiple regions on multiple axons. Not the case in the myelinating cell of the peripheral nervous system. So, only two types of cells we're going to talk about. Two types of glial cells in the peripheral nervous system. We have satellite cells, and we have neurolemmocytes. The other name for this that you will commonly find are called Schwann cells. So Schwann cell, neurolemmocyte, same deal. Again, these two are only found in the PNS, peripheral nervous system. So here is the spinal cord. Here we have the roots of a spinal nerve, or a nerve coming out of the spinal cord. This little structure here is called the posterior root ganglion. We'll go over all of this once we get into the section on the spinal cord. But I want you to know that in the posterior root ganglion, you will find collections of cell bodies of sensory neurons. So here we have the cell body of a sensory neuron. Notice this is a unipolar neuron. And we have these glial cells surrounding the outside of the soma, or the cell body. These are the satellite cells, small supporting cells that are covering the cell bodies of neurons in the peripheral nervous system. Right? Their job is to support, maintain, and keep these neurons nice and healthy. Also, notice here, we're in the peripheral nervous system. Each one of these little marshmallows is its own support cell. Right? These are the next type of cell we're going to talk about, called neurolemmocytes, also called Schwann cells. So remember, oligodendrocytes, many branch cells, one cell myelinating multiple axons. Here, with the Schwann cell, the neurolemmocyte, one cell myelinates one small section of one axon. Right, so they're responsible for myelination in the peripheral nervous system. Again, increases the speed of the action potential, and it allows it to occur much more efficiently, so it requires less energy. Same structure, or same function, I should say, as an oligodendrocyte. So this is the neuron, one neurolemmocyte, 2, 3, 4, 5, 6, 7, and you can see it has a nucleus, and notice it has this layers of the myelin sheath. What myelin actually is, it's a lipid, it's a fat. So you're going to find it in the phospholipid bilayer. So the way you could get lots and lots of myelin wrapped around one axon is you have lots and lots of cellular membrane of the neurolemmocytes. So let me just cut right to the picture here. This is an axon of a neuron, the peripheral nervous system. This is the neurolemmocyte, a regular sort of blob-looking cell, and it's just going to engulf or cover the axon, and then it's going to start spinning around and around and around and around, putting down layers of the plasma membrane, right? So that's the cytoplasm of the neurolemmocyte. This is the plasma membrane of the neurolemmocyte, and each time it spins around, it's going to add more and more and more layers of phospholipid bilayer of the plasma membrane. But remember, for these cells, you're going to get lots of myelin in the phospholipid bilayer. So the more layers of cell membrane you have, the more myelin you have. This is the electrical insulation around the axon. Okay. So this table here gives a very nice summary of the first part of this lecture. Definitely want to know the name and appearance and function of these types of cells. Astrocytes, ependymal cells, microglia, oligodendrocytes, you should know, yes, those are only in the central nervous system. Astrocytes, ependymal cells, microglia, oligodendrocytes, only found in the central nervous system. Satellite cells, Schwann cells, neurolemmocytes, only found in the peripheral nervous system. Also be aware of their function. Myelination, I talked a little bit about this, but let's continue, talk a little bit more. So what this does is it allows information to travel down the axon much more rapidly because of the presence of this lipid called myelin, right? Lots of myelin and lots of layers of plasma membrane makes the myelin sheath. In order for to have a healthy functioning nervous system, you need to have myelination of both the central and peripheral nervous system. So I talked a little bit about these figures. What this is showing you is a neurolemmocyte, but the same basic concept applies to the oligodendrocyte. Here, you still have, in the central nervous system, you'll still have many layers of plasma membrane. It's just that in the CNS, one oligodendrocyte can myelinate multiple axons, multiple portions of multiple axons. On the peripheral nervous system, one neurolemmocyte myelinates one small section of one axon. So the way that this works, more or less, is when you have this insulation, when you have myelin, the action potential does not have to travel down the entire axon in a linear fashion. What is going to happen is it's going to simply jump from here, boink, it's going to jump to this little space in between the neurolemmocytes, right? So these things are sometimes called neurofibril node, axonic nodes. The old name is the node of Ranvier. So instead of the action potential traveling all the way down, it will simply jump, boink, 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 from one of these nodes to another. So it will just hop from one to the next. So it's faster and it requires less energy. How exactly this is possible, I will save for physiology to understand all the details there. These are sometimes called saltatory jumps, which means they're simply going to jump from one node to the next. You might ask yourself, well, why not simply have one giant neurolemmocyte that runs the entire axon? That way you could simply jump from the very beginning to the very end. And the answer to that is, it would be real nice, but it doesn't work. These jumps can only travel a certain distance, which just so happens to be the exact width of one of these neurolemmocytes. So exactly how they function, what are the particulars of the action potential, how are they generated, how are they able to jump, all that is a subject for your physiology class. In this class, we're looking at the structure. Here in the peripheral nervous system, we can see we have neurolemmocytes, which are responsible for myelination. And in the central nervous system, we have the oligodendrocytes. Now, you should be able to look at this and tell me, without any labels, what part of the nervous system we're at. And you should realize that by looking at the neurons, you cannot tell. By looking at the neurons, you can't tell if you're, in this particular example, if you're in the CNS or PNS. But you can tell by looking at the glial cells. One cell myelinating multiple sections on multiple axons. That can only be an oligodendrocyte, which can only be in the central nervous system. Down here, one glial cell myelinating one small section of one axon, can only be a neurolemmocyte and can only be in the peripheral nervous system. Now, this next section, I just want you to be vaguely aware of. I'm not really going to go into crazy detail here, but understand that just because a neuron is damaged does not necessarily mean that it is irrevocably be destroyed. Some of you may have received an injury, and then you may have lost feeling in a particular area or a particular limb, or lost the ability to control something. And then like weeks or months later, you sort of regain control. You might say to yourself, well, how is that possible? If the neuron's dead, it's gone. Well, the answer is the neuron can be unable to perform its function in communicating information, but it doesn't necessarily mean that it's dead. So what you can have is damage to an axon, which then later is repaired by the neuron. And that's what this little section is. I'd like to explain how that can happen and what are the role of the neurolemmocytes. So in this example, right, by the way, what kind of neuron is this? Afferent? Efferent? Where is it in the body? What part of the nervous system is it in? All right. You should be able to answer these questions by looking simply at this figure. If you're not sure, talk to me during the Zoom session. So here we have a multipolar neuron, right? Its axon is extending out to, looks like a skeletal muscle fiber. And there's some sort of damage, right? The axon is cleaved, right? That doesn't necessarily mean it's going to lead to the death of the rest of the cell. In fact, the rest of the neuron is fine. In fact, the vast majority of the neuron is just fine. It's just that the axon has been damaged. But if that axon's broken, an action potential is not going to travel. So you're going to have paralysis of this muscle, right? You're going to get stimulation of this neuron, but you will not get contraction of the muscle. So what happens is the remaining portion of the axon will repair itself, while the part that's been separated from the cell is going to degenerate. Those glial cells, which have been myelinating the part that disintegrate, they kind of go back to their blob-like form. Now, this loss of structure, loss of the distal portion of the axon, the neurolemmocytes returning to their original state, creates a structure, right, called a regeneration tube. So what this does is it provides a pathway for the axon to regrow because it's being guided and being helped by the neuroglial cells. There's already a space created by the cavity left over from the older material that had degenerated. So the axon can regrow down the regeneration tube, get remyelinated in the process, and it may eventually reconnect to that skeletal muscle. Now, neurons are really, really, really long. So it's going to take a really long time to transport material from the cell body all the way down that axon. So this regrowth process is going to be slow. It's going to be slow. It's probably going to reconnect to the skeletal muscle in a way different than before. So it's not going to behave the same, right? It's going to be a little bit awkward, right? When people will get nerve damage and they have to learn to walk again, they're really having to recircuit their nervous system to make new neuronal pathways, new neuronal circuits. And it's a long, laborious process, and it is not a lot of fun. But if you can reestablish these connections, you can at least make a lot of progress. So just realize that nerve damage doesn't necessarily equate to nerve death. Right? Axons can be regrown, new connections can be made, new neuronal pathways can be worked out in the brain, but it is a very arduous process for anyone who's ever had to do it or has seen it or has helped somebody do it. So that's what's going on with axonal regeneration. Okay, let's talk a little bit about nerves. What exactly is a nerve? A nerve is a collection of axons in the peripheral nervous system. So you want this definition down exactly. A nerve is a collection of axons in the peripheral nervous system. Right? This is different from a tract, which we'll talk about in the next chapter. A tract is a collection of axons in the central nervous system. So if I use the term nerve, you should know I'm talking about a collection of axons that are going to be found in the peripheral nervous system. Let's see here. And we're going to have 3 layers of connective tissue surrounding them. These terms should look kind of familiar. Endoneurium, perineurium, epineurium, right? This is exactly like the structure we saw in muscle. Instead of endomesium, perimesium, epimesium, now we just call it neurium. So endoneurium, surprise, surprise, is areolar connective tissue, which is going to separate one myelinated axon from another, right? This helps prevent short circuiting, right? We didn't want one muscle fiber's action potential triggering the action potential of another muscle fiber. Well, we don't want the action potential of one axon triggering action potential in another axon, right? That would lead to chaos. Let's go to the picture. Here's an axon. It will be myelinated by what type of cells? Because this is a nerve, it's a collection of myelinated axons in the peripheral nervous system, because it's part of the peripheral nervous system, these must be neurolemmocytes. Right. That kind of connection, you definitely want to have just like clean. Right. A nerve means PNS, PNS means neurolemmocyte. So you do want to have that level of understanding. We have an axon, it's myelinated by a neurolemmocyte wrapped in endoneurium. A collection of myelinated axons taken together is a fascicle. Just like a bundle of skeletal muscle fibers taken together was a fascicle. One fascicle is separated by another fascicle by perineurium. Right? A collection of fascicles together is a nerve. The outside of the entire nerve is covered by epineurium. So if you remember your structure of your skeletal muscle cell, you already know the structure. With just a couple little tweaks, you already know the structure of a nerve. An axon carrying information away from the cell body. Axon covered by neurolemmocytes, by myelin, wrapped in endoneurium. A bunch of axons, a bunch of myelinated axons all together make a fascicle. Wrapped in perineurium, a bunch of fascicles together makes a nerve wrapped in epineurium. These cells still need nutrients, they still need oxygen, so you'll tend to have blood vessels traveling inside these large nerves. Now, one thing I should point out, the nerves in the body, nerves will carry information in both directions, both sensory and motor, right? A neuron only carries information in one direction. So what you're going to see is in a nerve, some information is afferent and some information is efferent. If you want to know what they look like under the electron microscope. Now, our last topic will be synapses. So we talked about synapses in the section on skeletal muscle, but that was specifically for a neuromuscular junction. However, you can have synapses between multiple different types of cells. Of particular interest is the synapse between one neuron and another neuron. And that leads us to some specific terminology. The presynaptic neuron, remember a synapse literally means a space. So the presynaptic neuron would be the neuron going into the synapse. So this is the space. You got one cell over here and one cell over there. So the presynaptic neuron, its axon is going to the synapse. On the other side, you have the postsynaptic neuron, the cell receiving the information. What part of a neuron receives information? Either the dendrite or the cell body. So let's go to the picture, right? Presynaptic neuron, the neuron before the synapse. Postsynaptic neuron, the neuron after the synapse. What's the synapse? The space in between the two. Information travels down the axon from the cell body of the presynaptic neuron. Right? That potential travels down. It will then cause calcium gates to open. You don't need to worry about that because it's physiology. Active potential travels down the axon, will lead to the synaptic vesicles fusing with the plasma membrane, releasing the neurotransmitter. The neurotransmitter diffuses across the synapse, binds a receptor, which will then eventually trigger a second action potential in our postsynaptic neuron. So exactly what's going on with the sodium and calcium channels, don't worry about that. Gated potentials, you don't need to worry about that. That's a topic for physiology. In this class, understand that when I talk about a presynaptic neuron, what part of a presynaptic neuron is going to be a part of a synapse? It has to be the axon. A postsynaptic neuron, the part involved in that synapse has to be either the dendrite or the cell body, because remember, information can only travel one direction, from the dendrite and cell body down the axon. Information or action potentials only travel in one direction. So if you remember the particulars about vesicles and neurotransmitters from the previous chapter, you're good. You just need to remember this terminology about post and presynaptic neuron. Now, something I've alluded to, but I haven't actually shown you, is a different type of synapse. It's called an electrical synapse. This is a chemical synapse because a chemical is carrying the information from one cell to the other. That chemical is the neurotransmitter. In an electrical synapse, we don't have a chemical. What happens is this change in membrane potential will simply travel from one cell through another cell, through a little hole, through a little porthole, through a gap junction. So here, when you have an active potential, you're going to have ions moving. More about that in physio. Ions can simply move from one cell through the gap junction into another cell, and then the signal can propagate. This is particularly important for cells that are really short, like smooth muscle and cardiac muscle. Cells that don't have a neuron going to each and every single cell like you can do with skeletal muscle, because those cells are really, really long. So you will find chemical synapses between neurons and skeletal muscle cells, between neurons and other neurons, and you will find electrical synapses between one smooth muscle cell and another smooth muscle cell, or perhaps between one cardiac muscle cell and another cardiac muscle cell. Here you have direct transfer, ions going through the gap junction from one cell into the other, allowing the action potential to pass from one cell to the other. Here in the chemical synapse, you have the action potential traveling down. It needs to be converted into a chemical signal, the neurotransmitter, which then spans the gap because the cells are not directly connected with each other, and bind a receptor, which then generates a second action potential on the second cell, the postsynaptic cell. So make sure you understand the differences. You already understand most of the chemical synapse, and I'm just throwing the electrical synapse in there and just giving you a few more terms. But hopefully this should be fairly straightforward. If not, review it in the book. Talk to me during the review session, because we will need to understand these two principles when we talk about the brain and the spinal cord, and again, when we start talking about the heart and the digestive system. And that brings us to the end of our section on neuronal tissue. Next time, we're going to start talking about the brain and spinal cord. Till then, take care.